So we're going to talk a little bit about some of the genetics aspects of Duchenne muscular dystrophy. And to start us off, we have Stan Nelson. He's a genetics professor at UCLA. And some of you might know him as the father of a famous person called Dylan. I met Dylan last night, so I'm a big fan of his. So welcome, Stan, and start us off.
Thank you, Anne. Yes, I'm mostly known as Dylan's dad. It's fair. Talking about some of the same issues that Pat had, pushing the button correctly. And will there be a timer on here? If there can be, that'd be great. You keep talking, keep me reined in. So, I'm a human geneticist, and I practice medical genetics in the clinic at UCLA, and we do genetics work trying to push the frontiers of how we might do genetics better, how we might find mutations better. In Duchenne, the disease gene was well known to be an X-linked recessive disease 30 years ago when the disease gene was cloned and identified. That is, females are typically carriers and don't manifest the overt full disease, and boys, their sons, can and have a 50-50% chance of manifesting the disease as indicated. That's right. I'll wait my hands for you. I'm going to talk this way. So that was well known, and this gave Ron Morton and Lou Kunkel and their teams good information about where roughly the disease gene landed. But most families actually look like the one down below, and similar to what Pat described, that is, no Duchenne is known to be running in the family, and there's an affected child who came out of the blue as far as the family is considered, and yet it's very clearly a genetic disorder. So I'm going to walk you through how that is, why that is, and a little bit on a number of different genetic issues. The human genome is vast. It's 3 billion letters. If you were to say this 1 second at a time for your whole life, you'd say it once in your whole life. And yet we can sequence the entire human genome in a few days routinely now with some of the new tools of modern genetics. Excuse me. There's about 24,000 genes. Duchenne is one of those genes, and it's located at the tip of the X chromosome on the short arm. It was discovered in 1986 by Lou Kunkel and his team. It immediately led to us having a much better sense of the full diagnosis and how to do carrier testing and know exactly what the transmission risk was in each individual family. It also provided amazing insight into related disorders, Duchenne and Becker muscular dystrophy, which actually leads to some of the key therapeutic strategies that Pat just touched upon. So it is the largest gene in the human body. It's encoded over 2.5 million of that 3 billion bases of the human genome on the X chromosome. And if you look at it in terms of the full genome sequence, it's plotted up above in the more black and white figure. So each little line is the business part, the exons of the gene, and the rest, the thin line, are the introns, and the parts that are removed before the RNA is translated into protein, the dystrophin protein. Most of us will think about the disease like this, though, and we'll just completely ignore that 99% of the gene that's the intron. So these are all the exons, and you'll hear over and over again at the conference about the 79 exons of the dystrophin gene. This is also one of those rare diseases where most of my medical genetics practice doesn't look like this and doesn't have the impact of exactly what a mutation looks like in terms of therapeutic strategies and importance to the individual patients in clinic. So knowing what the gene was, knowing that it was encoded by these 79 different exons over 2.5 million bases of sequence led to specific molecular diagnostics, but it also led Lou Kunkel and his team, Eric Hoffman, to identify what the protein, not sure what the protein is that's encoded. It's a long protein that's a long filament, and from that one can figure out where it's located. You can make a series of tools, antibody reagents, and see where in the cell it's located and what its function might be. So in normal muscle, this is a muscle fiber, on the left is a bone, then a tendon, then the muscle that you can actually see. You can't see much on me, of course, but in others you might see a little bit more. And each muscle is set into a series of bundles ending up to an individual muscle fiber, which is in the blue square down below. And each muscle fiber is a very, very long, multinucleated, meaning there's many, many different nuclei, cells that fuse together to make a long individual muscle fiber. And on the membrane of that muscle fiber is known to be where the dystrophin protein is localized, just underneath the membrane. So in this figure, the blue line is the muscle membrane, also called sarcolemma. And above it is outside the muscle, and below it is inside the muscle. And one analogy to think of for the function of the dystrophin protein is that it acts like a shock absorber, which is indicated by the wheel in the car below. So dystrophin would be the blue line below. So the left end of the dystrophin protein, the pink line, is like it's attached to the car wheel. And so that's binding to the actin, which is part of the motor inside the muscle. The middle part is a series of springs acting like a shock absorber. And the right side, which is also called the C-terminal end, is attached to the car in the model down below, or attached to a whole series of other proteins that are in the muscle membrane. And that this protein is critical to protect the muscle cell from contraction-induced damage as you go through into your life. So without it present, we get the constant damage to muscle fibers over time, and that leads to the phenotype that we see, the disease that we see, which is Duchenne muscular dystrophy. So this is now a slide of the muscle in cross-section, through this way and then stained. And in the upper left-hand corner in red is a dystrophin surrounding each individual muscle fiber in an unaffected child, and down below is Duchenne muscular dystrophy child showing no dystrophin protein staining. Up above is a healthy-looking muscle fiber. The muscle fibers all come together. They're pink and healthy and protected, and they're all roughly the same size. And down below, some of that muscle is degenerating and is various sizes, and it's being replaced by some fat and fibrosis, as what we're seeing histologically. We know that when this gene was cloned, it was a very big deal in human genetics. It identified not only the molecular basis of Duchenne, but also the molecular basis of the allelic disorder Becker muscular dystrophy. So on the left is Dylan at age 12 to 13 when he lost ambulation. This is fairly typical of Duchenne in this era, with chronic steroids being used fairly early on in life. And in contrast, the allelic disorder Becker muscular dystrophy is often not diagnosed until later in life, and many can still be walking in their 40s, 50s, or 60s. So indeed, it can be a very different disease. And indeed, they're both caused by mutations in the same gene. So that was figured out fairly early on to be due to the fact that mutations in dystrophin on the far right lead to no dystrophin whatsoever. And these are loss-of-function mutations, no functional protein. The middle two are two Becker children, which have smaller bands. The protein's a little bit shorter, a bit in the middle is missing, and there's less of it, as judged by the intensity of the bands on this chip. If you look at the same immunohistochemistry, which is the staining of the muscle in cross-section, up above is normal, down below is Duchenne, and in the middle is Becker muscular dystrophy, showing some small amount of dystrophin staining in the muscle, and that's characteristic of Becker muscular dystrophy. This is all relevant for how we, as medical geneticists and you as families, should be interpreting the genetic reports that you get. Sorry about that. So this is an alternate view of what the 79 exons look like. It's a reading frame map, and it tells you something very simple in terms of if you take a given exon out, have you affected the reading frame of the map? And we're going to come back to this in 1 second. But one of the things that was noted was if you had a deletion of exon 45, actually the single most common mutation that we see in Duchenne, it would be out of frame because the flat edge of one of the exons 44 does not match to a flat edge of 46, but instead is shifted. And so the 3-base genetic code is now shifted for the rest of the protein. It leads to no functional protein. In contrast, you can have a very large mutation in the middle, missing exon 17 through 48, and have a very mild phenotype because the flat edge of 16 matches to the flat edge of 49, and it creates a functional protein. Another way to look at that is these two are drawn out here with the original diagram that I showed you in terms of the dystrophin protein structure. The left end is the N-terminus. It's the actin binding domain, ABD here. The right end, in the orange, is where it associates with the protein complex to the right. And in the middle, all the blue is like the springs of the shock absorber. So that's normal up above. If there's a small deletion, the exon 45 deletion indicated in red, and then go down to the second one down, it stops the protein being manufactured in the middle, and that leads to the protein being degraded completely. So this is a non-functional protein completely. The second red line down below is this much larger deletion, and with that larger deletion, even though it's huge, much, much bigger, it affects the protein in a much more mild way. And that's because at exon, the exon in the teens, it's joining the exon in the 40s in frame, so it's possible to keep making the full protein, just missing that middle part, and that's highly functional. You wait for me, how much time? 1 minute? Oh, good Lord, okay. Okay, sorry. So the reading frame is the central issue that we always look at. One of the things that we will look at and interpret for every single mutation along the way is, does this mutation cause a change in the open reading frame? So when you discuss this with the geneticist, that's the key thing that we'll be looking for. And does this lead to no functional protein at the end of the day? It's led to the idea of exon skipping, and this slide illustrates that in terms of a language option. Most of the mutations, and most of the people in the audience will have mutations in the range of 45 to 55, because that's the mutation hotspot. Most people have multi-exon or single exon deletions. Those are the most common numerically. But point mutations and small deletions and duplications do exist. One of the things that's notable is that when we have small mutations, the amount of information that we have about some of these is less clear. That is, the exact functional impact for some of them is a bit less clear, and it does want us to do muscle biopsies in a subset of the individuals. The other areas to keep in mind are that mutations that are at the beginning of the protein and towards the end of the protein are a bit more difficult to fully interpret, and some in-frame mutations can still be quite severe, and actually some out-of-frame mutations can be a bit more mild than mutations in other parts of the gene. I'm going to just touch on two issues real quickly here. That is, for some where we never find a molecular diagnosis. This is one family where we, with all current modern molecular genetics, we couldn't find the mutation. Taking a muscle biopsy, doing an RNA sequencing analysis and whole genome sequencing analysis allowed us to combine those two powerful technologies to find the exact base pair. What happened in this family is it was a balanced inversion of genomic DNA, so it looked exactly the same for all sequencing and all of our molecular tests, and that inversion could lead us to make a family-specific PCR diagnostic for the family, and indeed that molecular probe could be useful for diagnostic testing, and we could do carrier testing in the mom and sister in this family and work out the full genetics in the family. So there's families in the room that have that issue. That's something that's worthwhile to talk about, and those are research protocols for the most part at this point. The other one I would just indicate, this final example is another instance where sometimes we ask for muscle biopsies still. So this is an individual with a deletion of 49 through part of 54. When the mutation breakpoint occurred within the middle of the exon, we didn't know what it looked like at the RNA level. So in this instance, doing RNA sequencing analysis, clarified that exon 54 is partly included into exon 55, and it has direct therapeutic implications about whether this child would be relevant for being on exon 55 skipping therapy. So in summary, everybody needs to know their molecular genetics. We can't always find it in everybody. It's relevant for not only to know how to give family counseling appropriately, but for some therapeutic trials at this instant. A portion, and a small portion, need molecular genetics to be done on a muscle biopsy at the end of the day, and there's tools to do those now. Thank you.